Thank you for joining us for today's Laboratory Tech Talk. In today's talk, we will review microbiological laboratory testing for validation of radiation sterilization, including test method validations, enumeration and characterization of bioburden, verification dose calculation, and test of sterility used to confirm the verification dose. Specific challenges encountered for large single-use bioprocessing systems will be detailed, which can significantly impact the timelines for the completion of radiation sterilization validations and dose audits. So first, let's start with the review of the microbiological testing used to validate the sterilization of a single-use bioprocessing system using radiation methods. The standard references for guidance on radiation sterilization validation and associated microbiological tests are listed here. ISO 11137-2 standard guides us with the process used to validate Method 1, Method 2, VDMAX 15, and VDMAX 25. ISO 13004 outlines additional VDMAX dose options from 17.5 kilogray up to 35 kilogray. For the microbial testing carried out in the radiation sterilization validation, we refer to ISO 11737-1. This standard covers bioburden testing, including the testing for inhibitory substances and bioburden recovery efficiency. It also gives guidance for product families and bioburden testing and data use and trending. ISO 11737-2 provides information on sterility testing, including the sterility method suitability, also known as bacteriostasis fungistasis, which is used to validate the sterility testing method. The microbial testing of a radiation sterilization validation occurs in 4 phases or stages. The first phase is test method validations. Test method validations ensure accurate microbiological test methods for the device, such as ensuring acceptable recoveries of microbes with the sample and the selected design of the test. Phase 2, enumeration and characterization of bioburden, is where determination of the number and types of bacteria and fungi contributed to the medical device as a result of the manufacturing process and raw materials. In phase 3, determination or calculation of verification dose, the bioburden enumeration data from phase 2 is used with the applicable table in the ISO standard to determine the verification dose range for phase 4. Phase 4, the verification dose experiment, is where samples of the product are irradiated at the verification dose range, and subsequently the samples are used to perform a test of sterility. Acceptable results of the test of sterility validate the sterility assurance level of 10 to the minus 6 for the minimum sterilization dose. With regard to phase 1, test method validations are an important part of the radiation sterilization validation, as they ensure that the results obtained are accurate and appropriate. There are 2 considerations for test method validations used in the radiation validation process: bioburden recovery efficiency testing and screening for inhibitory substances in both the bioburden and sterility tests. Test method validations are required by the standards and expected during device review and audits by regulatory and notified bodies. The bioburden test method validation is critical for the routine determination of a product's bioburden, as it ensures that the test method to be employed will give true data, essentially ensuring that acceptable recoveries are obtained and there isn't a gross underestimation of product bioburden. Organisms may be more or less recoverable from different materials or substrates, and this test measures and corrects for the recovery from the specific device being evaluated. Bioburden recovery efficiency testing validates the removal and culturing of organisms from the product and ensures accurate measurement of contamination on or in a sample. Bioburden recovery test method validations can be determined by either a native repetitive method, often used for products with consistent high levels of natural bioburden, or by inoculated method, which may be used for products with low levels of natural bioburden or those not suitable for testing by the exhaustive recovery method. Bioburden recovery efficiency testing is one part of establishing the bioburden test method, with screening for inhibitory substances being the other part. There are two inhibitory substance screening tests used in the radiation validation process. The first, the bioburden inhibitory substance screening, is a test that ensures that the product's presence in the extraction fluid does not give a reduced bioburden count. This test is a quantitative test resulting in a number of colony-forming units obtained and compared to control samples. The required outcome is that the growth recovered from samples containing product is not significantly different to that seen in samples containing no product or positive controls. Note the completion of inhibitory substance testing may not always be applicable, but should be considered. This consideration should be documented by the manufacturer and maintained in the quality system. The second inhibitory substance screening test is bacteriostasis fungistasis testing, or sterility test method suitability. This test ensures that the product presence in growth media does not create a falsely negative or no growth result. This is a qualitative test, meaning the presence or absence of growth, and is tested against several compendial organism strains. Required outcome for this test is that there is growth of the test organisms in the presence of product within 3 to 5 days of incubation. Bioburden testing is performed in accordance to guidance from ISO 11737-1, and the verification dose is most often determined from tables within ISO 11137 or ISO 13004. Again, this is a quantitative test, so we will have a specific number of microorganisms as a result, which is reported as colony-forming units. For a full radiation sterilization validation process, 10 units from each of 3 production lots are tested with the validated bioburden method. The bioburden determinations should be carried out on product that has undergone the manufacturing through final packaging process. The product must also be reflective of routine devices manufacturing and the contamination that may be present for the routine sterilization process. Bioburden testing or enumeration process is primarily performed by flushing, cutting up, or disassembling the device to place it into a sterile testing container. When cutting up and disassembling the device, extensive manipulation may be necessary to allow for placement of the sample into the test container. It is worth noting, the more manipulation, the greater the possibility of contamination during testing. A sterile rinsate solution is then added to the test container. Generally, suspension fluids such as buffered water or buffered water with a surfactant are used. The sample is extracted to remove microbes from the device. There are a variety of extraction methods which can be used individually or in combination based on material rigidity and design, such as sonication, mechanical shaking, vortexing, or stomaching. After the extraction process, the extract fluid may be divided for culturing of different organism types. The extract fluid will generally be assayed by either filtration method or pour plating method. For the filtration method, the extraction fluid is poured into a filter cup with a filter membrane in place. These are located on a filtration manifold. A vacuum is applied and the test fluid is suctioned through the filter membrane, trapping bacteria and fungi present on the filter membrane. The filter membrane is then placed on a growth-promoting agar for incubation. Note that pour plating is used where filtration method is not an option due to the product debris, product particulate, gels, or other items that may clog or degrade the filter membrane. Pour plating is carried out as follows. A portion of the extraction fluid is pipetted directly into a Petri plate. And then tempered growth-promoting agar is poured over the plated extract fluid. These are mixed and the agar is allowed to cool for solidification before incubation. Once cultured and enumerated, we can also characterize the type of organism present on the product. Characterization of the colony-forming units present on the device gives insights to the likely sources of bioburden and their potential resistance to sterilization. The identification of the organisms can also assist in understanding how to appropriately group products into families or with selection of a master product. Microbial characterization is also useful during investigation on exceeded bioburden limits or sterility failures. Most basic forms of identification are selective culturing, colony morphology, and gram staining. Selective culture uses selective media, heat shocking of extract, or incubation conditions to detect certain types of microorganisms. Colony morphology gives a visual description of the colony's growth characteristics, such as color, shape, consistency, etc. For example, the organism in the top left picture is cream, wrinkled, dry, with an irregular edge. The visual description helps to classify observed phenotypes over time. When coupled with further identification processes such as gram staining, it can give insight into the possible genus and species recovered. To obtain a higher confidence level match, genetic identification or MALDI-TOF identification testing can also be completed. Matrix-assisted laser desorption ionization time of flight, or MALDI-TOF, uses ribosomal proteins analyzed by mass spectrometry to compare spectra to a library of spectra from known organisms. In a full radiation validation consisting of 3 independent batches, the verification dose is set using the overall average bioburden. However, if any single batch out of 3 tested has an average of twice or more than the overall bioburden average, then that single batch average, i.e. the highest batch average bioburden, is used for dose setting. We take the overall bioburden average, or that highest bioburden average, and look it up in the corresponding table in ISO 11137-2 or ISO 13004 for the corresponding verification dose. Table 5 in 11137-2 gives us the values for method 1, table 9 for VDMAX 25. Note that there are additional tables in this standard that are available for other VDMAX doses. And ISO 13004 gives us alternative VDMAX doses from 17.5 to 35 kilogray. Once the verification dose range is determined from the bioburden results, samples are then irradiated at this range for the verification dose experiment. For method 1 and method 2, 100 samples are required for the verification dose study. For VDMAX methods, 10 samples are required for the verification dose study. Dosimeters are used to measure the dose provided to the samples. The verification dose range determined has a plus or minus 10% range. If the dose used is less than 90% of the result, or less than 90%, and the results of the sterility test are negative, the results are acceptable. Tests of sterility is performed for these samples, and results are evaluated according to the corresponding radiation standard. Again, the test of sterility is a qualitative test where we get a positive or growth or negative or no growth result. There are 3 acceptable methods within ISO 11737-2. The preferred would be the immersion test, where we directly transfer the sample into a liquid growth media. Extraction method is an alternative test where we can wash the product, followed by filtration of that wash liquid with subsequent immersion of the filter membrane into the growth media. Similarly, if the sample is a liquid, the filtering of that liquid can be performed and the membrane filter placed into growth media. If the extraction technique is used, extraction efficiency must be appropriate, and a risk assessment must be performed if utilizing the extraction method. Once there is a validated radiation sterilization process, dose audits are performed on a regular basis to ensure maintenance of the radiation validation. ISO 11137-2, section 10.1 states that dose audits are carried out to confirm the continued appropriateness of the sterilization dose. This dose audit confirms that bioburden is under control and that the sterilization dose continues to achieve the required sterility assurance level. Dose audits are required during not required during periods in which product is not produced. When infrequent production is performed, dose audits with each production batch may be appropriate. The frequency for carrying out dose audits is outlined in ISO 11137-1, section 12.1. Section 12.1.3.1 indicates that the interval of time between dose audits can either be 3 months or a different time interval can be selected based on a documented rationale. In section 12.1.3.3, it identifies that 12 months is the longest or maximum interval between dose audits. So what needs to be completed in a dose audit, or what is needed to complete a dose audit study? 10 samples are tested for bioburden regardless of the validation type, VD-Max, Method 1, Method 2A, or Method 2B. Following exposure to the verification dose, additional samples are tested for sterility. Depending on the validation type, there is a different number of samples that are tested. VD-Max requires 10 samples, Method 1 requires 100, Method 2A and 2B also require 100 samples. So what are the best practices for most devices for the bioburden test? We're typically performing testing on all portions of the device. Sample components are immersed in the extraction fluid or liquid, and the method of extraction, whether that's sonication or mechanical shaking, or a variety of other combinations of methods, yields consistent and acceptable recoveries. Agitation by sonication or mechanical shaking can aid in the removal of organisms. The test method would allow for detection of results below limits established for radiation validation type. For instance, VD-Max 25 has a requirement that the overall average bioburden is less than 1,000. Our method would need to detect well below that. Sterility, best practices. The sample would be able to be completely immersed in the liquid growth media or broth. These sterilized media is contained in the test vessel we intend to use for that test, which is usually a glass jar. The test vessel would also preferably be clear so that we can observe for growth throughout the duration of the incubation, which is 14 days. The sample is able to fit or be made to fit by either cutting or bending the sample to place it into the wide mouth container. If immersion of the sample is not possible, extraction might be used. And again, this requires risk assessment and an appropriate recovery efficiency. So what are the challenges for testing single-use bioprocessing systems? First would be the size of the devices. These large devices often are difficult to fit in the limited size containers that are available for laboratory use. They can be reused in the laboratory through autoclaving and autoclaving the media within those containers. There is increased manipulation if we are manipulating the device to fit it into those containers, which can lend to a greater potential for incidental contamination during the testing process and other health and safety issues for the technicians performing that testing. Limitations also exist with equipment. Laminar flow clean benches have specific sizes up to about 8 feet in length and specific depths as well. Incubators can be a limiting factor if we're filling large bioprocessing bags that are meant to hold, say, 500 liters of growth media for a cell and gene therapy product or pharmaceutical product. Those all would need to be fit into the incubator, and we need between 10 or 100 depending on the validation type. Additionally, sonicators and mechanical shakers have limitations to the size of items that can be placed in them or on top of them, as well as the weights associated with that. Large volumes of liquid growth media required to submerge or fill the device can create some workplace safety concerns, as they can become quite heavy, and if they are disassembled or cut up, it can lead to some repetitive stress or injuries to the hands through cutting those materials, which can be sometimes very durable. For large systems, the fluid path is often more achievable for testing. The fluid path may be the most critical for the sterile claim, but understand that if the sterilization validation is based off of the fluid path, the exterior likely has a different sterility assurance level or bioburden reduction occurring to that portion, but it doesn't have the same sterile claim as the fluid path. And it may not be known through this validation what that outside portion is if we're only testing the fluid path. The entire device, if we're testing it, often requires the use of a sample item portion or a simulated product, that being a smaller version of the actual product with all of the same components and materials, just at a smaller comparable size or type of components. We could also use large bags as test containers. These have some disadvantages in that they can be punctured by sharp objects within the test as well, whether there's needles or sharp pieces of plastic, they can puncture the bag. Similarly, bags can become quite heavy and difficult to move. The use of extensive manipulation and handling, again, to place the samples into a test container can increase the potential for contamination. And pouring of growth media into a test container, rather than merely transferring the sample into sterile media, can also potentially lend to contamination. It's important to note that if using a sample item portion or simulated product, that the bioburden may not be evenly distributed. If it is known that the bioburden is evenly distributed, the sample item portion that we're using could be used from any portion of the device. If it is known that it is not evenly distributed, the sample item portion could be selected from proportionally representative portions of the product materials, or if it is known and not evenly distributed, the sample item portion could be selected from a specific product portion that contains the most severe microbial challenge, that being numbers and types of organisms for the sterilization modality. If selecting the most severe microbial challenge for a sample item portion, the relationship between the bioburden of the SIP and that of the entire device should be established. And this is detailed in section 5.2 of ISO 11737-2. Also, we want to ensure that any simulated product does not allow for leaks similar to the actual product. It should go through all of those manufacturing steps that the actual product would go through. In ISO 11137-5, section 5.2.5, there is a test detailed, which is to ensure that the sample item portion selected is adequate. And this is done through one of two ways. Either there's a test of 20 unirradiated samples of the product, where the SIP with the sterility test method is performed. 17 of the 20 samples have to yield a positive result in that test to establish that is an adequate sample item portion, or 20 samples could be tested and yield a bioburden result of 1 CFU or more on at least 85% or of the 20 or more sample item portions. If this test does not work, a larger sample item portion would need to be selected, or the entire device would need to be used. For radiation, pre-cutting or disassembly of the device can be performed to reduce the manipulations required post-irradiation in our test of sterility. However, it's not acceptable if the preparation of that sample changes the magnitude of bioburden on the device, either reducing the bioburden or increasing the bioburden so that it is no longer representative of the product. It is also not allowed if it changes the chemical environment in the vicinity of the organisms, such as an anaerobically packaged device that is cut up and placed in a package with oxygen. This would no longer be representative of how bioburden would be presented in irradiation for the actual product. Additionally, inline filters can pose challenges for testing. They are made to trap organisms, and they can trap the organisms that we're using to validate the method of bioburden recovery, making it difficult to get acceptable recoveries. Additionally, they can impact the test for sterility method suitability in that they limit where the organisms can go throughout the system. They can be limited to just a portion of the device, which is not a full challenge of all sections of the device. They are best tested by destructive methods that require physical manipulation to essentially destroy the housing and then immerse the entire contents as well as the housing. Additional challenges are open-ended pathways, filling tubes or open-ended tubings on the system. If we're testing the fluid path of the device, obviously we can't have open ends, as the test media would just pour out of the end. Additionally, if we're using caps or fittings or clamps to close ends, the closure needs to be significant to not allow microbial ingress during the 14-day incubation of the sample. It is possible to use appropriate sterilized caps, provided they do give that closure that we need to ensure that the test is not getting contamination in during incubation and handling. Additional challenges from complexity of devices is that the more complex a device is and a large number of tubing pathways that need to be organized in the testing environment is an increased or prolonged exposure of the sample to the environment. Although it is a clean environment, there is still some potential of contamination when you're working in a clean room. So there is an increased risk of contamination the longer that sample is exposed for complex devices. Customized devices also, single-use systems also add variability. So for the validation of that product, perhaps a worst case product is developed to represent all of the variability that could occur within that portfolio of products that are sold. Complex systems can take additional time to fill or disassemble. This can affect the amount of time that the samples could be exposed in the clean environment, and the manipulation that needs to be performed to the test sample, increasing the potential for incidental contamination. And those customized single-use systems could add variability to the sterilization process and may require the creation of the simulated worst-case product that represents various configurations of the device. In general, When approaching sterilization validation of single-use systems, it's important to plan and communicate with the test laboratory to ensure that a strategy is developed for the bioburden and sterility test. When we do this and support these testing strategies, we optimize the validation timeline and also allows for coordination of supplies and resources to ensure that there are appropriate and adequate supplies to perform the testing when samples arrive, and that resources such as personnel and safety are established well before the samples arrive. This helps to ensure a safe and quality process for the lab work of these types of devices, and ensures that our technicians leave work the way they entered work unharmed. Thank you.